All right, welcome to the next podcast and in our look at DNA, its structure, and how it copies itself. This will be podcast 32B, and what we are going to look at in this podcast is the idea in general of DNA replication and how that would occur. We talked in the last podcast about how DNA is built, about its complementary nature, A's to T's, C's to G's, the sugars, the phosphates, and the bases found in nucleotides, and how many of these nucleotides connect together to make a big, long DNA molecule. We talked about the double helix, the two-stranded nature of DNA. So, what we're going to do is look at how the DNA molecule is built and the effect that how that allows for it to do what it does in terms of copying itself to give exact copies to offspring. Okay, so that said, I really do not. So let's do that. All right. So how exactly does this duplication happen? It is pretty simple on the surface of things. If you see a T, you need an A. That's pretty straightforward. So let's talk about it in the simplistic terms, and then we will get into the nitty-gritty of how it actually might occur at the molecular level. So if this is our parent DNA molecule, we have a cell, it's got this strand of DNA. If this cell wants to make two cells, it wants to have baby cells, it wants to go from being one cell to being two cells, it needs to give each daughter cell an exact copy of that DNA, or else this daughter cell will not be able to function properly. So we must copy this, we must copy it exactly. So the way that occurs is first the parent molecule, the parent cell has to split its molecule of DNA down the middle along the hydrogen bonds. Remember, there are hydrogen bonds connecting these bases, A to T, C to G. They're hydrogen bonds. They're relatively weak and can be pretty easily separated. Okay, so we open that up. Then we have enzymes in place in the nucleus of the cell that can come in and grab T from the cytoplasmic or nuclear soup and add it opposite this A G opposite this C, an A opposite this T, a T opposite this A, a C opposite this G, and do the same for the other side, an A opposite T, C opposite G, T to A to T, G to C, and so on. And in the end of it, you have brand new DNA molecules, two of them that are absolutely identical to this one that we started with. They are identical, okay? And then we can give this one to one daughter cell and give this one to the other, okay? That is the basic idea of DNA replication. Done. Except for this is AP biology, and we can't be done with just that. We have to talk about some more nitty-gritty details. I do want you to understand this term. Understand that if we go back and look, notice that this new DNA molecule we have here and this one here is actually made up of half old DNA, original parent DNA, and half brand new DNA, half new, half old, okay? That term for that of having each new molecule made up of an old strand and a new strand is called semi-conservative, meaning we have conserved half of the old strand in the new strand, okay? And the semi-conservative model of DNA replication is shown here. These were other ideas that turned out not to be true. This is the one. We have the original parent, and then we have this one old and one new making up the daughters, and this shows how it would go on from generation to generation. Okay? But semi-conservative is the word you need to take away from that. Um, Now, how this replication is actually carried out, there is a whole team of enzymes that are going to catalyze the reactions necessary to make the DNA, allow the DNA to copy itself. There are some things we should keep in mind as we are talking about DNA copying itself. In a human, there are 46 DNA molecules in each of your cells, except for blood cells, which are just sacs of hemoglobin with no nucleus, no DNA. But there are 46 DNA molecules in every single cell in your body. 46 double helices in every single cell, 46 chromosomes. If you add up all the base pairs, all the A's to T's and C's to G's, and those DNA molecules, those 46 DNA molecules, that's about 6 billion base pairs. 6 billion. You need to understand how much information that is. That is like 900 of your AP biology books, which I know you haven't used, but if you'll recall it, well, here it is. It's about 900 of these, 900 of those to print out. It would take 900 of those book pages of those books to print out all the A's, T's, C's, and G's that make up your genetic code. Yet, your cell can copy all of that information in just a few hours, okay? And it is very, very good at it. Imagine copying 900 biology books worth of information in just a few hours and making only one error per billion nucleotides. That's staggering that it can be that error-free, and thank goodness it is, because every error in the DNA can lead to a catastrophic problem. It doesn't necessarily have to, but it can. Okay? So, what you need to remember is that everything we were about to talk about, replication, you need to think back to mitosis for a minute, what we learned with that. The S phase during interphase is where what we're about to talk about, replication of DNA, occurs. That is the time period. So if this is our cell cycle, okay, and this little part is mitosis, prophase, anaphase, all that. All of this, from here to there, again, all of that is interphase. And if you'll remember, interphase is basically divided into three parts, G1, S, and G2. It is this part here where the DNA is copying itself, getting ready to divide during mitosis. So this is all happening during this point, okay? Basically what we're doing when we replicate is we're going to take those single-stranded chromosomes we start with in G1, and we are going to make an exact copy of them and join them at a centromere. Stop that right there. And so they become double-stranded. So we're going from single-stranded chromosomes to double-stranded chromosomes. And in this, once they're double-stranded, we call each of those sister chromatids until we separate them again at the end of mitosis, and we go back to calling them chromosomes. So that's just reminding you how all of this fits in to mitosis. It is very important to understand that. Now, you need, as we begin talking about replication, you remember we numbered the carbons. You have to be able to do that. It really does matter. So remember, we got the 1', we got the 2', we got the 3', we got the 4', and we got the 5'. Of all of those primes, which is important, this one, oh God, no, Coleman, I'm an idiot. Man, I can't make it go away. Hold it. Hold on. I'm killing myself. You're killing me, Coleman. All right. Erase all ink on slide. All right. If we go back, 1, 2, 3, there we go. Okay. This one is the important one, and this one is the important one. All the rest of these are unimportant. 3' and 5'. It really does matter. Okay. Now, some other things we're going to talk about is that replication in prokaryotes and eukaryotes. First of all, you need to understand that I don't care if you're a bacterium, a prokaryote, or everything else, a eukaryote. DNA is the genetic material. It is one way that all life is connected, very fundamentally. And replication, making copies of the DNA, whether you're a prokaryote or a eukaryote, is really quite similar. They're really basically the same, but there are some differences that we will hopefully have time to mention. One of those differences is how replication begins, the origins of replication, meaning the place on the chromosome where we start copying the DNA, okay? A bacterial chromosome, if you'll remember, a bacterial chromosome is really just one molecule. That's all any bacteria has, and it is one molecule of DNA, and it's in the shape of a ring. So basically we take our DNA molecule and we just curve it around on itself and connect it and make a ring out of it. That is what bacteria or prokaryotes have, one ring-shaped DNA molecule. And this ring-shaped DNA molecule has one place along it somewhere where we will start replicating. Maybe right there is the start or the origin of replication. And replication will proceed in this direction and in that direction around the length of that until ultimately you end up with two DNA molecules at the end. Okay, eukaryotic chromosomes are different. If you'll recall, a eukaryotic chromosome is the DNA is not in a ring, it is in a straight line. Sorry, that's a really lame double helix, but whatever. Okay, and in fact, there are multiple start points. An origin could be here, an origin could be here, an origin could be here. And replication will go this way, and 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 this way. In other words, it goes from both directions from each point. So what you're doing when you have multiple origins of replication is you're making replication faster, because you're going to start right here and go out in both directions, and they're going to meet. As this one is going this way, we're going to meet in the middle. So we are going to make replication go much more quickly rather than just having, like, for example, suppose this is our DNA molecule and we started just at the beginning, the one end of the strand, and went all the way to the other one. That would take much longer because you'd have to do one from one place, the entire strand. Here we're starting many places and we're going to do a bunch of it all at the same time. Okay? So that's an important difference between prokaryotes and eukaryotes in terms of replication. So an origin of replication, then, is a region on the DNA that has a specific sequence of nucleotides, and that sequence is recognized by certain proteins, enzymes perhaps, that will recognize that sequence and attach to the DNA at that point so that they can start copying the DNA. Okay? So when proteins, like for example, maybe right there is an origin of replication. Maybe there's a particular sequence of TA, TA, TAA, something like that. Okay? And there are proteins that recognize that genetic sequence, so they latch on right there and start to open up the DNA strand, and they begin to copy it at that point. When they open up the DNA strand, we call that place where it's opened a little replication bubble, because it kind of spreads apart at that point. And here you can see a much nicer drawing. Okay, so this is the DNA strand. These dark blue represent the original parent strands of DNA. This is where the sequence of DNA was that told the little proteins where to latch on, and this light blue is the new DNA getting laid down, the new strands getting made, and we're proceeding to copy out from the origin. But the same thing is happening here and here as well. So These are all origins of replication, and as you can see, the bubbles that are created get larger until they meet, and you eventually have two complete daughter DNA molecules. Okay? So once a bubble is created, then the replication is going to proceed in both directions until the entire molecule is copied. Okay, we can have many thousands of replication bubbles in a eukaryotic cell all happening at the same time. So replication would proceed in both directions from that point of origin. That's just a repeat. Now, this term, replication fork. Okay, if this is my DNA molecule and I'm opening it up, if you can look, okay, right here is kind of a Y shape. That is the replication fork, and that's where the action is actually happening. There's a replication fork on this side as well, and we're copying into the Y of the replication fork. New strands are being elongated at those points. That's where all the activity is. Okay? Some other concepts we need before we get into the actual mechanics of all this is the term anti-parallel. Anti-parallel means that the sugar-phosphate backbones run in opposite directions. Now, gosh, Lord have mercy. What does that mean? Again, remember that 3' and 5' matter. Okay? 3' and 5' matter. The phosphate group is going to be attached to this one for this nucleotide, so this would represent a nucleotide, and then if this is the next nucleotide, the next phosphate group for the next nucleotide would be attached to the 3' here. That's why they are important. Okay? What this means is that the DNA shows polarity. It has two different ends. If this is a DNA molecule, okay, and those represent the connected bases, A, T, C, G here, this end might be 5' with the 3' exposed at the bottom. If that is the... let me go back. Back, go. Okay. Actually, maybe in forward. Here we go. Okay. If we connect them, this end might be 5', this end might be 3', but these DNA molecules are put together when you've got your double helix. One moment, please. They are put together like this. If my two pens, the pointy ends are here, the DNA molecules are not put together like that. They're put together like this. So if this is 5' to 3', then this is 3' up here and 5' down here. Okay? This is going to be actually quite important. Okay? Let's see if I can get you. Okay? There, this is the diagram I wanted you to see, right here. So if we put our DNA molecule together like this, not only are A's and T's, C's and G's complementary to each other, but they are flipped upside down, the whole strands relative to each other. So if the 3' end, 3' carbon is exposed here at this end, and the 5' carbon is what is exposed at the top here, and this one is flipped upside down in reverse. 3' is up here, 5' carbon is down here. Okay? That's going to be really, really important for how replication occurs. The enzyme that allows DNA to copy itself, it is called DNA polymerase. We're going to talk quite a bit about it, but it can only add nucleotides to a pre-existing 3' end. It cannot hang a nucleotide onto a 5' end. It just doesn't work that way. The enzyme can't work that way. So what we need to do is think of 3' carbons as hooks for other DNA molecules, and we can't hang a nucleotide onto the DNA without a hook. So DNA can only grow in the 3' direction, never, ever in the 5'. Okay? And that is going to matter. Okay? So I'm going to stop there, and then we will talk exactly about how the enzymes work to build a DNA molecule. Thanks for watching. I'll catch you again in 32C. See you later.